Hey, everybody. Here again today with Dr. Rodney Nash to talk about homogenization and single cell isolation. Welcome, Dr. Nash. How you doing today?
Hi, Pete. Thank you for having me.
Thanks for being here. Rodney, so I guess the quick question is homogenizing versus single cell isolation. Can you tell me briefly, in layman's terms, what the difference is between regular homogenization, which Omni's been doing for 50 years, and single cell isolation?
Absolutely, Pete. Traditionally, when one thinks of homogenization, they think of lysing cells, complete destruction of tissue, lysing cells, and releasing whatever the analyte for downstream analysis. This could be nucleic acid, this could be proteins. Whereas with single cell homogenization, what we're doing is we're actually just dissociating cells. We're not destroying them. Cells are still intact, markers are still there. And uh, downstream analysis could also be performed if desired at whichever point.
So, if I understand you correctly, traditional homogenization would be basically high impact, mechanical, ultrasonic, bead beating, where you potentially where you're definitely disrupting a sample, but potentially breaking open cells that you would not necessarily want to do with single cell isolation. Is that correct?
Absolutely, that's correct. With the higher impact homogenization, yes, the goal is to release the analyte in that particular step for downstream analysis. Whereas with single cell analysis, what we're looking at is we're trying to just separate the cells from the tissue and look at markers without actually destroying the actual host cell.
So you mentioned the analyte. I have a question about enzymatic portions of homogenization or single cell isolation. What role does enzyme play from or enzymatic digestion or enzymatic lysing play in single cell isolation, both good and potentially not so good? Can you give me some information on that?
Well, good is it actually frees single cells. Uh, Bad is that typically it does it at a cost of altering markers. So these markers are important, just like how we recognize each other by our face typically. These markers are how we identify cells. For instance, if it's a B or T cell, it could be CD4, CD8, or CD19. If it's cleaved, we see cells that are present, but we can't identify the cell. So this is not good downstream. Additionally, other things such as the transcriptome, the actual expression of genes, becomes altered when cells get stressed by enzyme and other types of chemicals such as that.
Right. So in a solution for our customers, we would want to strike a balance between enzymes that would free up single cells, but not um, overly disrupting or stressing those cells at the same time. Would that be correct?
Absolutely, absolutely, Pete. Enzymes are not necessarily bad. It just depends on which enzymes you're using and how much and at which stage are they introduced into the workflow. So absolutely, they can be used as again with the solution that Omni has. It's very low enzymatic activity, and we're able to actually separate cells, keeping them intact and keeping all of the actual desired markers present on the surface.
That's really, really interesting, and I think our customers are going to be very interested in hearing from you further in the future. So I'm looking forward to having more conversations with you, as well as the rest of the Omni team having conversations with you about this topic. Rodney, Dr. Nash, thanks for joining us today.
Thank you again for having me. I look forward also to the future.